HIV is the virus that HIV is the virus that causes AIDS. Here are shown white blood cells in the bloodstream. HIV enters the cell. It's a virus, and it begins to proliferate. But as it proliferates, it has an enzyme that helps it reproduce its genetic material. And when it reproduces, it makes mistakes. So every virus in the cell is actually a different virus. These viruses are then released from the cell, and some go on to infect other cells. When these cells are infected, it replicates again, producing different types of viruses. And this process goes on and on. And this is how HIV propagates its infection throughout an individual. Now, one of the major strategies for treating people with AIDS is to introduce one of, or a set of different drugs, shown here on the right, that can kill the virus and prevent its replication. But this presents a problem where we create resistance to the drugs. So when the drug's administered, it goes into the white blood cells and other cells, of course. And now it's going to prevent viral replication and inhibit the virus. So what happens is that most of the virus goes away. But in some cases, as shown here for the red HIV virus left in the cell, this virus had accumulated mutations that allow it to persist in the cells. And even if it is inhibited mildly, it still survives. And then this virus is able to replicate and propagate the cells. And as before, when this is secreted from the cell, it can go in and infect other cells, even in the presence of the drug now. And then the virus continues to proliferate. This is one of the major problems with therapy for AIDS. So, over the past 3 decades, scientists from numbers of different institutions and pharmaceutical companies have helped to develop approximately 27 drugs that are called antiretroviral drugs that are used to fight AIDS. And often these are used in combination. This is a great accomplishment. However, what we see over time is that some strains of HIV are becoming resistant to one or more of these drugs, and these can be transmitted between people. Or you can have what's called de novo resistance, where new resistance is acquired to a drug in a person. So as we use these drugs to treat people across the globe, a lot of times we're giving drugs to people that are already resistant to the drug. Thus, we're wasting billions of dollars of drugs with ineffective treatment. Now, our lab has come up with a strategy to help address this issue. We call it goMAP HIV. And it's a simple web tool. And what we do is we know the mutations that occur in the virus because we've built a giant database of sequences of these viruses. And we know each country that the viral infection took place. Thus, we can predict the resistance rates in different countries of the world to any of the drugs by just analyzing sequences that came from that country. Now, shown here is an atlas of the world with a heat map to a particular HIV drug. The darker colors indicate high frequencies of resistance, with the darkest color indicating greater than 20% of the patient's sample being resistant, and the white color being no resistance. As you can see, for this particular drug, the resistance is central to Northern Africa, but there are regions of the world that are appearing that are starting to show resistance. This is a common profile for many of the HIV drugs, except we see different centers where some resistance is centered in, say, South America or the Americas. some in Europe, and some resistance profiles show a broad-based distribution. So what we aim to do here is to help change how HIV drugs are used in the world by creating a resistance report that can be used. Shown here is South America with resistance to a particular HIV drug. And the coloring scheme is different before and is shown in the upper left, where red now indicates greater than 20% resistance. And as you look on the top axis of this table, these are the names of different HIV drugs, and they're colored differently based on their class. So if we come and look at Argentina, for these 20 or 20 plus drugs, you can see for the ones on the left where there's lots of red, there's high resistance to this set of drugs, which are called protease inhibitors. But there are other drugs like those on the right where there are little resistance. So if you were treating a patient in Argentina, you would try to steer clear of these drugs on the left side in favor of those on the right side. So it's a very simple concept. But now, for any country, a country can collect sequence data, which could be just directly imported into our database, and we can make an annual or quarterly report of what the resistance profile is for that country, so that the policymakers can decide which drugs are more effective to use in those countries. So this is the concept of goMAP. goMAP was produced by the Schiller Lab at the University of Nevada, Las Vegas. It's been funded by the National Institutes of Health, and we have a publication shown here, and web links to the project are shown here. If you're interested in helping to fund a staff member to help maintain this database, and so that we can provide these reports to many countries of the world, please contact Martin Schiller at UNLV.edu. Thank you.